So let's say you found the skeleton. How would you tell it's a dinosaur? You personally, how would you decide?
Well, interesting question. I don't know. I'd have to do some research on that.
Well, as you might be able to tell, I did not properly set up my intro video. So you guys got to see a quarter of the intro video. Sorry about that, everybody. I will fix that after we go off air. However, I am in fact here with Otangelo, Grasso, and Sphincter of Doom, who is, of course, my Navy buddy. So, Otangelo, why don't you say hi to everybody?
Yes, hello everybody. It's nice to be at your channel, Dapper, and thank you very much.
All right, and take that, Ian Chen. Otangelo Grasso did show up. I had a very good feeling that he would. So, mostly because we had several emails about it over the course of the last week or so. So, basically, this is a stream, and it is titled Falsificationism and the Philosophy of Science, because Otangelo, during our debate, I realized that we were having different conceptions as to how science proceeds and how it is properly to be undertaken. And so I thought that we should have a conversation about that. And my friend, I'm just going to call you Josh. Is that a problem?
That's fine.
Okay, because I'm not going to say Sphincter of Doom every time.
That's fine.
Okay, so, and my friend Josh here also had some ideas that he wanted to input there. And so, what I'm going to do is I'm going to give a quick little, talk, I guess, about why I think falsificationism is so important and how it contrasts with confirmationism, and why it is that I think that you might be working under a confirmationist paradigm. And so then I want to turn it over to you, and you can give a quick talk, and I'm not timing it, but try not to go for 20 minutes or something, and then we'll just kind of open it up to a sort of free-flowing thing. And before I really get started, I want to say that thank you to Scott Duke, who just gave a $15 super chat, which is very generous. He says, "I've been enjoying your content and your participation in other streams at Dapper Dino. Keep up the great work." Thank you, Scott. I really appreciate that. And those super chats really do help. Every single one is very, very much appreciated. So I'll just start with my little thing here. So for a while in the history of science, the scientific method had been somewhat established. The idea of making an observation, coming up with a hypothesis, coming up with ways to test the hypothesis, and then seeing if your predictions bore out, has been the core of science basically since the very beginning of the modern era. And I'm saying modern era in the sense of, a historian. So the modern era goes back at least into the beginning of the 19th century, and in many cases you would push it back into the 18th century too. But one thing that was important is that Karl Popper, who was a 20th century author, he made sure that science largely adopted a falsificationist view. So the goal of science, according to Popper, and now according to basically all philosophers of science who have any, sway in the scientific world, the goal of science is not to demonstrate something is true or to confirm a hypothesis. Ultimately, science is in the business of falsifying or disconfirming hypotheses. And the reason for this is confirmation is really easy to get. So, for instance, let's say I have a hypothesis that water causes measles. And I check this by going to measles patients and seeing if they have water in their blood. And every time it turns out, yep, they do. That's confirmation. I predicted that every time. But you could disconfirm this by checking people who don't have measles. And if they have similar amounts of water in their bodies, well then it doesn't seem like it's an important factor. And so you can disconfirm the hypothesis. Now, the part of science that really, really made this clear to Popper was psychology, and also to some extent part of history. So Popper noticed that despite the fact that Marxism had already been falsified in terms of its historical predictions, people had managed to reinterpret it in ways that made it completely impossible to falsify. Everything that could possibly happen in history could be interpreted by Marxists as in some way confirming Marxist theory. And also that problem happened in psychology with Jung and Freud's ideas about the human mind, which were competing, but also were both confirmed by literally every observation. But this is something that has extended to all of science. So now in science, what we try to do is we find a falsification for a hypothesis. And only when we can't do we then give that hypothesis tentative approval. And so now I would like to turn it over to Otangelo. And if you have any questions you want clarified, feel free to ask. And then just, have your own understanding of falsificationism and confirmationism and the philosophy of science and whatnot.
Yes, thank you, Deborah. Well, we had, since our last debate, which we had, I've moved a little bit forward in my understanding of the whole thing. And you brought forward 2 examples, which you said, well, these are systems that evolved and got irreducibly complex. And you said, in that case, the entire hypothesis or the framework of irreducible complexity has been falsified. Now, as I said there, well, since I have never heard any ID proponent to bring forward these 2 examples, I cannot tell you if I agree with them or not, but I will investigate it further and then I will give you an answer. And to be honest with you, the thing about HIV, it is very complex. You need to study it for some time to understand what goes on in regards to infection and HIV and so forth. And I have relied a little bit of other people which have told me about what they directed me in some sense how to respond to this challenge. And I think that my concept may be that there is a clear drawing line between something is irreducibly complex and it cannot evolve. Maybe it is a little bit blurred. And I think that Levy, in his last book, The Edge of Evolution, he brings some more clarification into this when he says that there can be up to 2 mutations in regards of malaria, and he makes some calculations and so forth. So it is not absolutely clear drawing line. This is irreducibly complex and it cannot evolve. And I think there is a certain, how can I say?
Okay, so Otangelo, real quick, everyone's having a bit of trouble understanding. Is there a different mic you can use or anything?
My microphone?
Yeah, is there a different microphone you could use or something like that? Maybe try unplugging it and plugging it back in.
Okay, let me try to plug in again and see if it's getting better, okay?
Okay. And sorry, everybody, the audio was a little bit cleaner earlier, and I'm not sure what the difference is or what's going on. I know that Otangelo has come on and had much better audio before, so I suspect there is some kind of...
Is it better now?
No, it's the same. Did you unplug your camera or your microphone?
No. I just changed channels here. Let me check it. Just a moment. Is it better now?
Much better.
Oh, yes.
That is much better. Okay, so one thing I want to point out is that this is really more about the difference between falsificationism and confirmationism in science, rather than about irreducible complexity per se. So, I mean, I do think that it's important because that was sort of the topic that brought this up, but
Well, my problem initially was to think, when you said, oh, these 2 examples which I am bringing forward, they are irreducibly complex.
Mm-hmm.
And I mean, I had not the tools at the moment to refute the claim because I didn't study these systems. But I mean, there was, how can I say, it was difficult to me to accept it because I can clearly see so many systems, they are irreducibly complex, and over years I've never seen any evolution proponent to refute these systems as being able to evolve, and that has come very clear to me. And there I said, well, I cannot fathom how these two systems eventually can actually be irreducibly complex, and therefore you can use inductive reasoning and generalize and say, oh, in that sense, there has to be some way for all these systems also to be able to evolve. In special, if we consider that irreducible complexity doesn't only hold in regards or as a contrast to evolution, but as well in regards of the origin of life, where there was no evolution, and I regard the cell as irreducibly complex.
Okay. So let's suppose, because we're trying to hammer down on the philosophy of science aspect here, let's suppose that there is a system which is irreducibly complex according to Behe's definition, which is the one you gave in your debate, and we know it has evolved. Let's just say, for the sake of argument, that those two things are true. Does that defeat the hypothesis that irreducibly complex systems are a problem for evolution because they can't evolve?
Yeah, I think that my position maybe has changed since our debate, Dapper, and I am working on this to understand, well, if one example actually meets the standard of falsification, then the concept has to be modified.
Okay. Now, I don't know if you saw it, but I actually put out I think about an 18-minute video on the topic of the philosophy of falsificationism as it regards science. Was it? I think it came out yesterday.
Yeah,
sorry.
I've seen it today.
Okay.
Before our debate, I watched it.
Oh, awesome. I didn't. Oh, thank you. I'm actually a little surprised. Oh, you know what? Now I remember you commented on something there. So yeah. Okay. Well, thank you. So the reason that I wanted to have this discussion is because the sort of counter you gave to the idea that a irreducibly complex system had been shown to evolve was to simply say that, well, if it can evolve, then it was never irreducibly complex in the first place. Now,
as I said, Dapper, I also thought about this, and during this interval since our debate, I had this interaction with someone which was helping me in regards of the HIV example, and then he brought up that Behe softened a little bit the concept and said, well, there are actually some systems where there is evolution, but that doesn't really falsify the concept of irreducible complexity. So there is a certain range where evolution is possible.
Okay. Now, so that's an interesting thing, because what we're getting at here is one of the responses that is acceptable in terms of science for a hypothesis that has been falsified, which is to say, or at least been apparently falsified. And so there are two major responses that you can take, and then I think we'll let Josh jump in here. So the first response is to say, okay, well, perhaps one of the background assumptions that wasn't explicitly formulated is faulty. Like, for instance, maybe the tools I use to measure the effect are actually inadequate. And so I need a different tool to actually find out what's actually going on here. Maybe that will, show that my hypothesis is no longer disconfirmed or falsified. Or another one is to slightly modify the hypothesis so that it gives different predictions, but ones that are still testable. But the important thing for either one is that we still be able to make a testable prediction that we then go and test. So, for instance, if there is, according to Behe, some systems which are evolvable that meet his definition, the thing that he needs to do, and that I don't think he has, but I've got to be honest, I haven't read a whole lot of him past Darwin's Black Box, and to be honest, it's probably been 5 or 6 years since I've read Darwin's Black Box, so, I might be a little behind on my Behe, is what I'm trying to say there. But as far as I know, he hasn't given any way to make new predictions that are both distinct from the old ones and also falsifiable. Would you agree that that would be an important thing to do in order to save the hypothesis of irreducible complexity?
Well, I don't know. I don't think that he has reformulated the first one. I think there are 4 versions which he changed a little bit from the 1996, the first version in Black Box. There are 4 versions, but I don't think he has changed the version after he wrote up the book The Edge of Evolution.
Okay. Well, then I think I would have probably read the most recent, or maybe the next to most recent edition. Which edition was it that had the chimpanzee on it?
I don't know.
Oh well, don't worry about it. It's probably not that important. But would you agree that the ability to make different predictions that are also testable is an important factor to changing your hypothesis to accommodate what otherwise would be a complete falsification?
Sorry, I didn't understand your question.
Okay. No, don't worry about it. I'll try to rephrase it. Let's see. Okay, so let's say I have an idea, and it has been falsified.
Okay.
And I still think that my idea is pretty good, and I think there's something to it. But I know that the simple form that I gave didn't work out. So instead, I modify my idea a little bit, and now it gives new predictions that so now it's a new hypothesis, but it's based on my old one, and it's now giving new predictions that I can test. And what was a falsification for my previous version of this hypothesis is no longer a hypothesis. But the important bit is that I have to give new testable predictions that can falsify my new hypothesis. Would you agree that that's an important part of how science needs to proceed if a hypothesis is falsified, but you still kind of want to work with it?
Yes, I agree with you. Now, I did read something that I would like to read to you that is basically in the formulation of the irreducible complex argument, which is not from Behe. And that is, let me check here, the principle of evolutionary continuity, succinctly formulated by Albert Leininger in his biochemistry textbook. An adaptation that does not increase the fitness is no longer selected for and eventually gets lost in the evolution. In the current view, only those adaptations that effectively decrease the fitness end up getting lost. Hence, any evolutionary scenario has to invoke at each and every step only such intermediate states that are functionally useful, or at least not harmful. And that correlates with what Behe says here: the removal of any one of the parts causes the system to effectively cease functioning. So this from Leininger, this correlates with what Behe is saying. If you have an intermediate product in a biosynthesis pathway, or if you have a modification of an enzyme that becomes non-functional, then it will not be selected. It will be discarded by the organism.
But I think that is ignoring the possibility that in quote-unquote intermediate stage may have a different function.
Well, regardless of its function, I'm not sure how that relates to the philosophy of science in terms of falsificationism. And I guess, Josh, what did you want to say about that? Because I know that you had some stuff you had set up.
Yeah, I mean, so the problem with confirmationalism is like you highlighted, is that you can go looking for data that accommodates your prediction, but you have to. But it's the central idea of falsificationism is that you can't know if you're right if you can't recognize when you're wrong. And like I know that the all swans are black or all apples are red is a common one, but you can go a little further in like something functional, like, and this is just because I'm a big fan of this particular show. I have a rock. It keeps tigers away. You don't see any tigers around, do you? I mean, that's confirmationally that hypothesis is demonstrated. But to test it, you have to go and find a tiger and see what happens. And then
do not test this hypothesis, people. Just accept that it's false and leave it alone. But yeah, keep going, Josh. Sorry, I just wanted to put a public safety announcement.
I wasn't genuinely suggesting that people find your local zoo. But then, okay, if you saw that it in fact did not keep tigers away, you could refine your argument. Well, this actually keeps Siberian tigers away, or it keeps cheetahs away, or you could refine it to something. and test it, but ultimately you still have to test it. And to test it, you have to your test has to allow for the possibility of being wrong. And if it doesn't, then you have a bad test. And if your hypothesis doesn't allow for being wrong, then you can't know if you're right, even if you are. And that's the biggest problem with confirmationalism.
So I guess, because I want to get Otangelo in here, so I'd like to ask him a quick question, because I know that he would really like to talk about irreducible complexity, and I'm okay with that as long as we have it in terms of talking about how it fits into the falsificationism paradigm in science. So, given that people have at least made the claim that seemingly irreducibly complex systems have evolved or are evolvable, actually both claims are made. Now, and assuming that those papers are making those claims are reasonably well supported, which the scientific community generally accepts that they are, that would falsify the basic hypothesis, which says that system meets the Behe version of IC, therefore IC as a problem for evolution is defeated. But if you want to have a new, broader range of things that might technically meet the definition, but some of them are evolvable and some aren't, is there a way before we do the tests to see whether or not it's evolvable, to predict which ones are likely to be evolvable and which ones are unlikely to be evolvable? Because that would be a new hypothesis. Because if you could make that prediction, then that would be a new acceptable hypothesis that science could test.
Well, the question is, if you have a system which becomes irreducibly complex, but which is shown that it evolved, and you don't have other systems which are irreducibly complex, but you have not shown that they could have evolved. So, I mean, that is possible. And then you have to ask, is it justified to use inductive reasoning in order, if you have falsified one claim, that you can generalize it to other systems and say, well, in that case, the concept has been falsified generally and it has to be modified?
So the problem is that if you're going to make the claim that irreducible complexity disallows for evolution in a particular extent, if you show that that isn't the case, you either have to refine your definition of irreducible complexity or refine your hypothesis with regards to irreducible complexity in evolution, which I believe he actually has in his one of more recent books, where he says evolution can allow for some diversification, but it doesn't fully explain it. But that still is that's a new hypothesis. So how do you test that then?
Well, I see it that way. When we say evolution, we have to actually clarify what we mean by evolution. And we all know that evolution to a certain extent occurs. And we know that there are a wide variety of dogs which have a wolf as a common ancestor. So we have to ask, where does that variation come from? And of course, that I think that mutations and natural selection plays a role. But there are scientists which also say that adaptation, which would be a kind of a microevolution, is actually a pre-programmed process already inbuilt into the organism, where animals, they have already, since the beginning, the ability to change, to speciate to a certain extent, and that would be a pre-programmed adaptation or evolution. But there is the third thing, which is the real part, which is in dispute, which is how do we explain the origin of big changes and organismal complexity and architecture. And that's where the big dispute is. And there are those which say, oh, small steps will lead to big steps, and micro will lead to macro evolution. And that is a very complex issue. And and also some people, they ask me, where do you put the drawing line between micro and macro? That is also a difficult question. And I think it derives from the fact that the mechanisms which actually explain organismal complexity, they are very varied, and it's not only genetic information which plays a role, but also epigenetic information. So that all the parts which are involved in organismal complexity building, they have to be included in any explanation. And then the big question is, can we explain all the biodiversity by natural mechanisms, or had to be intelligence involved?
Oh, okay, but Otangelo, I feel like I haven't received an answer to my question about what new hypothesis, because remember, all of those are interesting discussions, but they're not falsificationism and the philosophy of science.
Well,
So,
I think that one of the core issues, Josh, is what I just outlined, which is not something which is an invention of Behe, but it is the fact that each single step of mutations has to be either neutral, or it has to have a positive new trait in order to be selected for.
okay,
That is not a claim which came from Behe. This is from
Okay,
other
that
sources.
still doesn't answer the question.
let me rephrase the question. Maybe that'll help. Okay, the question is, assuming that the current definition, the definition that Behe gives of irreducible complexity, and so given that definition and the hypothesis, systems that meet this definition cannot evolve, and then given, and we're just assuming it, the observation that some of them have evolved and many can, or some can evolve and some have evolved, we now need to modify the hypothesis in order to make it a viable hypothesis again, or we can just discard it.
Okay.
How would you modify that specific hypothesis in a way that it gives new predictions that are now testable again, so we can go out and run a new test? What new version of that hypothesis that is falsifiable would you like to use going forward?
Well, maybe it should be elucidated what mechanism exactly was involved. You have several. You have mutations by natural selection, you have drift, you have gene flow. I don't know if you can extend it to epigenetic mechanisms, if they can evolve. It would have to be discovered in regards of that particular system, what was actually the exact mechanism which permitted that system to evolve or to change, and then eventually from there on start and modify the previous formula.
Okay,
So
maybe you can ask the question, Josh. I don't know.
So we're not disputing that. Yes, we would have to have a more sophisticated. You would have to have a more sophisticated hypothesis regarding the relationship between irreducible complexity and evolution. We're asking what that hypothesis would be. Right now you're describing that we would need that, and no one in this room I think is disputing that. But what would be an example of that hypothesis?
I don't know how I could answer that question at that point, Josh. I think that the basic issue here is that a system which is irreducibly complex cannot emerge by slight modifications. I mean, this is pretty hard to falsify it, because there are many examples which I have given, which Behe has given, which are out there, and they are conceptually. I think that evolution by unguided mechanisms simply is not the correct explanation of the origin of these systems.
Okay, well, I'll use an example maybe to help with the whole why I feel like that betrays a misunderstanding of falsificationism. There was a famous example of the difference between falsificationism and confirmationism is the thought that all swans are white, right? So for thousands of years, no one outside of Australia had ever seen a black swan, because, as it turns out, black swans were originally endemic to Australia. They didn't exist anywhere else. And since really no one from outside of Australia was going to Australia, at least in terms of people who were formally scientifically describing animals, who were mostly centered in Europe, the idea was all swans were white. And every time someone went out and saw a swan and it was white, that helped confirm the idea. But the minute that people got to Australia and saw their first black swan, that completely ruined the idea that all swans are white, because it only takes one black swan to overturn that idea. Now, let's say we're going to go back in time to before any, person who wasn't Australian or from a nearby island had seen a black swan. So we're back in time, we're in Europe, and we think all swans are white. And for some reason, God miraculously comes down and tells me, hey, there are exactly 84 million swans in the world. I don't know why he tells me. It's weird, but for some reason, God just really wanted me to know exactly how many swans there were. And for some reason, it was 84 million. Just picking a number. Okay. How many black swans do I need to find in order to know that at least not all swans are black? In that case. Now that I know that there's a whole bunch of them where I don't know their color, how many black swans do I need to find to know that some swans are black?
Yeah, just one.
Just one. So, if I just find one, even though there are a bunch of other untested swans that I've never seen, I know not all swans are white. Similarly, if it is the case that any system that meets the definition for irreducibly complex, if any one system has been found to be either evolvable or to in fact have evolved, then no matter how many other currently unexplained systems there are that also meet the definition, they cannot be a problem for evolution simply for being irreducibly complex. Now, that holds open the possibility that a different reason might be found for why they can't evolve. But what I'm asking you is, if that were the case, and I would submit to you that it is the case many times over, that we have found both actual evidence of the evolution of these systems and also very plausible evolutionary pathways for others of them, and since either of those completely falsifies the original hypothesis, you would need to come up with a new definition that would be testable. Now, I recognize that you might not have that handy, but you at least recognize why it seems to most, I'll say, secular scientists or mainstream scientists, that one, the hypothesis has been overturned and falsified, and two, there does not currently seem to be a modification that would allow the hypothesis back into consideration in science.
Well, first of all, I think there are people, and I think they have a point here when they say, I don't like that concept of irreducible complexity. I prefer to analyze each system one by one, individually, rather than putting a hypothesis on the table, which you can use in a varied way, from even a DNA molecule, which is composed of 3 parts, to a ribosome, which requires many parts, to a cell, which is another dimension of irreducible. So what I want to say with this, the concept can be basically applied to many different things, even to an ecosystem or an energy cycle, which is interdependent and irreducibly complex. So maybe rather than apply just one hypothesis and then use it in a very, very varied and general sense, it could be eventually better than we look to the systems one by one and then ask, could that specific system evolve or not? If not, why not?
Well, that's fine, but what you're doing there is actually testing evolution per se. You're not testing irreducible complexity. So it's fine to do that, right?
Yes.
But there's 2 problems that we have here. Having a bunch of systems that are not currently explained by evolution is not a falsification. You actually would have to find a system that you can but that like you can actually demonstrate with very high confidence could not possibly evolve. And irreducible complexity as a mechanism for proving, well, I'm using proving, colloquially. Using irreducible complexity, for one thing, cannot do that anymore because we already have that falsified. So if you want to be able to say a system cannot evolve, you need a new hypothesis that would be able to strongly predict which systems would be impossible and how to demonstrate that they would be impossible to evolve. Is there any attempt in the intelligent design, or more broadly the creationist, whether old or young Earth creationist, to actually come up with such a hypothesis? Because I'm not aware of one, but then I have read far from everything that every creationist has ever written, so maybe it's out there.
Well, I think that, first of all, it is not even necessary that we falsify the claim of irreducible complexity. I think just a thought experiment will illustrate why that the concept that something has to be designed and could not be the product of unguided natural mechanisms, you can come to that conclusion just by making a thought experiment without any experimentation or falsification at all.
I mean, you can have thought experiments about all sorts of things, but science isn't based simply on thought experiments.
Yes, but Josh and Pepper, as you both know, I am a proponent of intelligent design with the end goal to point to God. That is my and confirm creationism as a superior explanatory framework rather than natural mechanisms to explain origins and the natural world. So that is my end goal. And if I can reach that goal by experimentation and by falsification or only through philosophy and thought experiments, that doesn't matter to me. What matters to me is to have an argument which confirms what I believe to be true. And it's interesting that even Einstein thought that experimentation would not be necessary.
But well, first of all, Einstein made predictions that were verified by experimental observations. In fact, I mentioned that in my video.
Yes, but he made them just through his mathematics and through
right. That's how he made.
reality.
Right, that's how he made the hypothesis and the predictions. But people didn't simply accept his predictions until they were actually verified. Because until the actual eclipse, and I think it was.
Well, okay, but I can give you an example, a prime example actually, where I would say this is something which will never be falsified in regards of irreducible. You can call it however you want, but I would say irreducible complexity is a good framework to say it, and this is in regards of the origin of the first cell, the barrier from the non-life to life. And I would say there is a barrier. When you go lower than that threshold, then you will not have life. And that is irreducible.
I'm not sure what that hypothesis is.
It is precisely the hypothesis which Behe gives, that if you have not all parts together in order to have a minimal cell, it will be not functional. And if you remove just one of all the minimal proteins required, you will not have a fully working, living, and self-replicating cell.
Oh, okay. But so you said something interesting, and it's been pointed out to me, and I want to dig into that. You said, and I believe this is a direct quote, "I just want an argument which confirms what I believe." Now, the problem that I have there is that is the exact opposite of how science proceeds. And I understand the desire to have such an argument. It's always nice to have, something that helps you feel more confident in a closely held belief. But the problem is that what science wants to do is find ways to show that what you believe is wrong. And only when you fail at doing that, after trying rigorously, is it reasonable to then conclude that maybe you're right.
But look about the entire scientific framework, Depper. Dobansky, he said, nothing makes sense in biology except through evolution.
Yeah.
And there is no alternative explanation in science to evolution. There is none. You have only that framework, and
that conclusion came from having ruled out several other possibilities. It wasn't just pulled from the ether.
Well, I disagree with you in regards of that, Josh, because we know that since the 19th century and Thomas Huxley, when he came up with the X Club, they promoted materialism for over 20 years in the Royal Society, and since then, the predominant philosophical view is philosophical naturalism, where you cannot put even God into the door as a hypothesis and mention it as a possible explanation of origins. And so that makes basically also naturalistic explanations unfalsifiable, because you have no alternative. You don't give even the chance to have design or creation as an alternative to materialistic claims.
All right. Now, I want to respond to that, but first, we have a €5 super chat from Zach Brannigan, who I used to mispronounce as Zap Brannigan because I've watched a lot of Futurama. So I'm going to try and do this in the voice of Zap Brannigan. Dapper, this is just to reward your patience. I'd have probably lost it by now. And that's my really, really bad Zap Brannigan impression, so I apologize to everyone who had to hear that. But, you know. Well, so let's see. So the science does not involve a philosophical naturalism. It involves a methodological naturalism. But
the entire, sorry, let me interrupt you on this. You're wrong on that. The entire scientific framework is based on philosophical naturalism, where design is excluded a priori.
But design is okay. Let me try to get out what I was going to say, and then I will explain why I feel like that is not necessarily a problem, even if the explanation for some things turns out to be miracle. Okay, so because science is in the business of trying to figure out if ideas are wrong, and only after failing to do so, bothering to accept them, one of the dependent parts about that, this is prior assumptions in science, right? One of them is that previous results indicate actual consistent workings of the universe, and that they can be replicated in the future according to the same consistent operating principles, essentially, of the universe, wherever those might come from. And the reason that science cannot accept as science, a supernatural explanation for a particular phenomenon is simply that doing that removes the possibility of being able to make predictions from previous results about new results. Because if a miracle occurs, we cannot predict whether or not it will occur again. And if it does occur consistently, it would be indistinguishable from just the normal operating of the universe. So we wouldn't see it as miraculous, even if for some reason it were just that God was doing a miracle every time you did a particular kind of experiment. So, but I don't actually see that as a problem for detecting design or falsifying evolution. Because the thing is, one, we have known instances of design which are not supernatural, and we can conduct tests to detect design in some things. So for instance, a simple test to detect design in, say, a man-made object would be to go find an engineer designing such an object. That would be easy. Or if we could find, in fact, some way, or we could simply point out that we have no plausible mechanism without design for a particular object to come into being. And we have no examples of similar objects coming into being without design. Now, hold on. Now, I could grant you that this might actually be a problem that we could hold out for abiogenesis, because it is not well explained by science right now. And so,
not at all.
If we found some problem for abiogenesis that was completely defeating, that would then make it somewhat reasonable to say something funny must be going on. And I would say in that case that God might be, some kind of God at least, might be not a crazy option.
And that's the point, Pepper. Is there a framework where science says, well, if that is evidenced in the cell, then we have a conclusive viewpoint or shift of paradigm where we will move from naturalistic explanations to design explanations. Now, let me make that distinction, which I have made many times before. We have two different questions. Science asks two different questions. One is in operational science, where I agree that we should only use methodological naturalism, where we ask how things operate. What is the function of X? So we will make tests, and then we will find out, oh, the protein has that function. We will not put supernatural explanations into this. But when it comes to ask how did things actually in the past originate, that is a different question. And there I see the problem where science is not permitting design as a possible explanation in contrast to evolution or naturalistic explanations in general.
Well, science has not reached anything conclusively on the origin of life, and that's however life originated has no bearing on merits or demerits of evolution. And science, it's not that science
Yes,
isn't
of course.
allowing design, it's just that there hasn't been an experiment that has allowed design to be properly tested.
Okay.
And that's
Can I?
and one of the bigger issues I find is that the supernatural is kind of outside the purview of science. That doesn't mean that the supernatural doesn't exist. It just means because science can only test things naturally. That's just.
I would like to answer to that.
Let's.
I have answered to this many times, and I agree with you. We cannot expect, if creation of life was a one-time event in the past, then we cannot expect that God will agree to demonstrate to a scientist in the laboratory that he will repeat the feat which happened in the past. That will not happen. But we know by repeated experience that we as intelligent beings, we can instantiate blueprints, machines, production lines, factories, computers, turbines, transistors. And these are all things which we see in life in an analogous manner. We see genomes storing information, directing the making of machines which are proteins, which line up into production lines, which are metabolic networks. We know that genes, they operate like a computer. They have a hard disk, which is DNA. The cell is a chemical factory which manufactures the basic building blocks of life. We know that the cell basically works like a factory because it manufactures all the things which it requires itself. So we have a clear analogy here of things that we know that intelligence is capable of doing. And we see all these things in the cell in an analogous way, but also I would say that, there are many science papers which say that cells are factories in a literal sense, not just as an analogy, and molecular machines are machines because they operate literally as machines also in an analogous way. And when you have in 70 years of experiments attempting to recreate the origin of life, they have not come even close to try to understand how the basic building blocks could have emerged prebiotically. Then you have to ask, well, when will science come to the conclusion that natural mechanisms and explanations are simply an inadequate explanation to explain the origin of life? And it seems in the current framework that answer will never be answered, in a sense that they will say, okay, we have really reached the point where we have to say abiogenesis is a failure, there has to be a different explanation, and then look somewhere else. And the problem is, as I said before, that design is not considered as an equally possible explanation, and that should, in my understanding, change.
Okay, well, let me...
Go ahead.
So, one... I'm going to try and bring this back around to the philosophy, because we've been spending a lot of time talking about, gee whiz, this is cool or complicated, and that's not really important to the discussion. So
yes, but I am addressing the in my understanding, a wrong philosophical framework of current science today, where it should be expanded in regards of origins and questions of how things came to be, to reconsider also not a specific deity, but just a designer intelligence as a possible and plausible causal mechanism of things that we observe.
Okay,
in
let me
nature.
ask a simple question. If something in life wasn't designed, how would you know?
Well, in over 70 years, science has tried to explain how the basic building blocks could have emerged prebiotically, and there is
Otangelo,
a
you're answering a different question.
No. I'm answering your question. And
what was my question?
But how could it be confirmed that natural explanations meet the standard and are confirmed?
Not quite. My question was very specific. It is, let's suppose that a particular system in a biological organism was not designed. If that were the case, how would you know?
If you see evolution, for example, or if you see unguided mechanisms creating life in the lab, or even making the basic building blocks of life.
Okay.
The two alternatives to design are either chance, physical necessity, or evolution.
I'm not sure how that's 3 and not 1, but okay. So,
well, in regards of the origin of life, you have unguided random events, chemical interactions, physical necessity, that it couldn't be differently than there it is, and then finally intelligent intervention. That are, in my understanding, the range of possible explanations.
Okay. So, what I'm trying to get at is, currently, I'm only aware of a small handful of hypotheses for intelligent design that meet the qualification of being falsifiable. Irreducible complexity is one of them. Actually, it might be the only one I can really think of that I really do think is falsifiable.
Well, the other one is a specified complex information, and if you can demonstrate that kind of instructional assembly information could originate by natural means, and that is far from being demonstrated that it is possible. And then there is also the other problem of the origin of the genetic code, which is an assignment of meaning of codons to amino acids. And then you have to demonstrate that it is in principle possible that non-intelligent mechanisms can do assignment of meaning.
Okay, so your claim is, so let me try to make this, I'm trying to make sure I get it. I want to understand. So the claim is right now, if we can find a natural mechanism that can create some specified trait, and it's natural, then specified complexity, or specified, I'm sorry, what was the term that you used? I want to make sure I get it right.
Specified complexity, or you can call it instructional assembly information to make specific devices. And I mean, the analogy here is, I, as a machine designer, I sit at the drawing table, and then I have the instruction from my boss to make a machine, and then I have to make the precise blueprint to make that device. And the analogy is in regards of the information stored in the genome through the nucleotide sequences, which specify the sequence of amino acids to make proteins. If it could be demonstrated that this sequence, in this instructional assembly sequence, could originate by natural means, then it would be substantiated that material natural mechanisms are possible.
Okay, but that's not.
Well, I want to get to what the possible or falsification is, but first I want to read a super chat from Jerry Pena, who says, Otangelo demands abio be answered, but cancer research has much more research dedicated and can't find a cure yet either. Okay. I want to get this hypothesis down. So, if we could find,
can I quickly answer to this? I hear that several times that people ask, well, what is the use of intelligent design, because it doesn't have any practical application to improve our lives. And my question to this is, it is actually much more important than that. It substantiates a worldview, which is that a creator is behind and on the bottom of reality. And once this is substantiated, that it shapes the worldview of people, and they will build their entire life based on their worldview, and that shapes their morality, their expectations in life, meaning, moral standards, and so forth. So it is very important.
I know it. I want to address the engineering analogy as an engineer. So the problem I have with that analogy is you're essentially saying that because we know of artificially or designed instances of various mechanisms, you're suggesting that everything else must be designed and couldn't occur by like natural means. But there aren't bridges that exist that just occurred by natural forces. There are so just because
not everything. Okay,
that's what you're saying.
No. I was very specific. I was very specific. I said instructional complex information or complex specified assembly information to make proteins and cells, everything which is required in the cell. So that is one of the key legs of intelligent design, and the other is irreducible complexity.
Okay, so I want to see if I can get a handle on the falsification of specified complexity, because that is your claim is the two big hypotheses are irreducible complexity and specified complexity. So if
these are two, there are others.
Okay.
Okay.
Well, we'll take these two for now. So
okay.
In terms of falsificationism, I'm getting as a hypothesis, if a protein, say, the novel protein can be found to have evolved and it performs a specific function.
Now, you have to start from zero. It's not an evolution problem. It is a problem of getting the first proteome and the first set of about 1,300 proteins from scratch, from zero.
Okay.
That's the challenge.
So, if I can say, go ahead and say, a randomly assembled protein, if any randomly assembled protein can be shown to have a function in a cell, that would falsify specified complexity, would it not?
Yes, you have a huge sequence space, and the most that space you need to find a sequence which would become functional, which would permit an amino acid, a polypeptide to fold into a form which then would permit that protein to have a specific function, like topoisomerase, like helicase, like.
Wait, does it have to have a particular function, or does it just have to have a function that is identifiable?
It needs to have a function which is used in the cell.
Okay. But throwing together random proteins has resulted in functional proteins in organisms in experiments. That has already been done. It's a small portion of them, but randomly assembled proteins made from randomly generated genetic code that is inserted into cells can produce function in cells that is beneficial to the cell.
The problem is much bigger, Dapper. You have also an irreducible complex information system, which is consisting of DNA, which is the hard disk, then the information, which is the sequence of nucleotides. Then you require the genetic code, which is the translation from codons to amino acids, and then there is also the information transmission system, which requires proteins. So you have basically four components which have to be there. Otherwise, you do not have
hold on a second.
What you want.
Hold on a second. This is the problem I think that I'm having with you. It is, first I say, hey, would X falsify this hypothesis? And you say yes. Then I point out. Hold on. Let me finish. I've been very good about not interrupting.
Okay. Yeah. Sure.
Okay. And then I say, well, X is observed, and then you say, okay, yeah, but. So unless you're going to modify specifically the specified complexity hypothesis, could you at least admit that that hypothesis, as such, has been falsified? Because it's great to give me a different hypothesis that might be demonstrating IC.
I will address precisely what you are saying right now.
Okay.
So I'm asking you, the smallest ribozyme which we know of has 124 RNAs. Okay. So if you do not have that size, you will have no function. So you have a random sort of RNAs around. Let's suppose even that there are the 4 which are used in life, and you shuffle them to have a functional sequence. So the number of possible sequences is in the order of 10 to the 50th power.
Okay.
That is in the realm of the absolutely impossible.
Oh, okay, but I'm trying to get the hypothesis here. So you said that specified complexity would be instructions that produce a particular protein that performs a function in a cell. Correct. And those things cannot arise by chance.
Correct, yes.
Okay. Would an experiment involving shoving completely random chunks of DNA of a size sufficient to produce a protein, starting with a non-random start codon, I'll grant you, and ending with a non-random stop codon, again, I'll grant you. But in between, everything there is completely random. Then doing this to a large population of cells and having any of those proteins come out as functional, would that be a falsification of specified complexity as a hypothesis?
You would have to have the 20 amino acids that we are talking here about a sequence to have a protein, or to have like a ribosomal RNA, which would have about that size as well. You would have these monomers just laying around randomly, and you have to shuffle them, these monomers, to then have a functional sequence.
So is that a no or a yes?
Sorry, to what?
Okay, I'll ask again, and I would really like a yes or a no. Okay? Just a yes or a no.
Okay.
Would taking a randomly generated genetic sequence, or sorry, not a random, thousands or perhaps even millions of randomly generated genetic sequences, and inserting them into cells in such a way that those cells express that sequence as a protein, and then finding that any number of them actually perform functions in the cell, would that falsify specified complexity as a hypothesis, yes or no?
No, because you do not have cells at that stage.
But oh my goodness. Okay, hold on. I need to Josh, you go ahead.
Okay, so as I understand it, specified complexity requires that protein to be expressed in some way. So you have to have this, and the protein expressed has to be usable in a cell. And if it doesn't, then like specified complexity says that it wouldn't.
We can make a very
complexity seems, in my understanding, to presume the existence of a cell to test whether or not the protein is useful to the cell.
Well, we can make a very crude experiment. It's like a lottery, where you put even millions of 4 different letters into the lottery, and then play it enough to see what it would take to have a useful sequence, which would eventually then permit to have a functional protein or a ribozyme.
Does specified complexity require the existence of cells, since you have to test whether or not something is useful to a cell for the claim that specified complexity has that randomly generated proteins cannot be useful to a cell? Yes or no?
I mean, it doesn't require a cell, but if you want to use a cell as an...
Well, how can you test whether or not a randomly generated protein is usable for a cell if you don't have a cell in which to test the usability of that protein?
Okay, we are basically making a thought experiment here, Josh. That is, we know that proteins, they have all a specified complex sequence which is required in order to get the fold which permits functionality.
No, that's the hypothesis. We don't know that. That's your hypothesis. We cannot say we know the hypothesis. I'm trying very, very hard, Otangelo, to try to figure out how this hypothesis is supposed to work in a falsifiable manner. I'm really trying very hard. I think Josh can probably agree, and I think the chat can probably agree.
Yeah,
okay.
Yeah, we will go.
I would like to state my understanding of this hypothesis, and then you can tell me if I got it wrong or if I got it right. Okay. So, proteins, in order to perform functions, require a particular shape and chemical activity based on that function. Is that a good start right there?
Correct.
Okay.
Yes, correct.
These functions are performed inside living cells.
Okay.
Is that also a necessary part of this?
Yes, of course.
Now, these particular folds and functions are specified genetically.
Correct.
And the genetic information required to make a protein that functions cannot arise randomly.
That is our case.
Okay. And now, is there something about this hypothesis that I am missing?
No.
Okay, so would it be a reasonable prediction to say any amount of random DNA used to, and this is at the outset, we're starting out with it randomized, we're purposefully randomizing the DNA, that any amount of it inserted into any number of cells would only result in non-functional proteins? Would that be a reasonable hypothesis?
Yes.
Okay, so if I did that experiment, and I found a non-zero number of proteins resulting from this experiment that did have function, I have falsified the hypothesis.
Correct.
Okay, but that's already happened.
No.
All right. So the previous example that Dapper gave
has not happened
regarding randomly generated a protein synthesized or expressed from a randomly sequence of DNA that then did result in a functional protein within a cell. How does that not falsify structural complexity?
No, Dapper said if you find an example, then it is falsified. But I don't know of any experiment which has shown that this would be possible.
Didn't Dapper
if you
refer
want
to it? I guess we didn't refer to the specific paper.
We can just make the statistical analysis, and it has been done by Douglas X in 2004, and he said in the sequence space to have even a minimal protein is 10 to the 74th power. That is the number of atoms in the entire universe. How do you think this is a plausible scenario?
If I understand that paper correctly, it is not the random chance, the chance that a protein expressed from a random sequence of DNA occurs. That's the chance that a protein spontaneously forms randomly through, without ribosomes or an expression of DNA, which is a very different claim. If I understand,
we are talking about abiogenesis where you didn't have all this machinery. You have to think that what is your alternative? You have there basically the first problem is you have.
You're again. I feel like there's either a misunderstanding on our part or on your part in conveying what you mean by structural complexity, because you're saying that, as I understand,
I can try to explain it again to you.
Dapper's explanation of structural complexity, to which you did not dispute any part of it, included having the machinery in place, and now you're saying that now we don't have the machinery. We have all the machinery in place. Okay, so you have to shuffle in the regards of a minimal protein sequence of, let's suppose, 120 amino acids. You can make the calculations. I guess it is about in 10 to the 50th power. And you know the size of that number. Well, you can calculate it. Calculations for what, though? What are you calculating?
You calculate that in each amino acid, you have 20 amino acids to select from, and they have to be left-handed. Okay? And you have a sequence of about 120.
Where did we get to isomerism in this? I am confused. Could you step back a second and say what you said again? Maybe I missed something.
You require a sequence of a minimal protein of about 120 amino acids. Okay?
That's not what a protein is. A protein is at minimum 50 amino acids.
Okay, let's even take that a minimum is 50 amino acids. Okay, so what you require is that in each of the positions you have to select from 20 amino acids which are used in life. Are we agreeing on that?
Used in definitely of life that we know it. Yes.
Well, we are working
Let's go with the 20. That's as fine a number.
Well, I don't know of any life form which doesn't use 20 or less. I don't know. So we have to work with.
Go for it.
So we have to work with what we know. So at each position you have 20 different amino acids to select from, and you have to sort them out from left and right-handed amino acids, because only left-handed amino acids are used in life. Correct? So actually, you have to select from 40 different amino acids.
Wait. How does that? I don't follow that last part. Maybe I'm missing something.
Okay, you know that amino acids they have two different homochiral forms. They can be left-handed and right-handed. Correct?
Yes.
Okay, so life only uses 100% left-handed amino acids. Okay.
Mm-hmm.
So you have to select. There is only in prebiotic life there is only a racemic mixture of right and left-handed amino acids laying around. There is no selection only of left-handed amino acids. Okay.
You're describing the fact that we don't know why only left-handed amino acids are used in life.
No, left-handed amino acids. Yeah.
What did I say? Right? I meant to say that.
Yeah. Okay, no
sorry.
problem.
But I think we're still getting kind of away from the question
there's
of how do we falsify structural complexity.
There's a few things here.
I am giving you a real-life example of what the challenge is, actually, so you can understand what we are talking about. It is not only about the sequence, which must be just right, and I already answered this. To have even a protein of 50 amino acids, you have 1 to 10 to the, I guess now, to the 20th power. So there is a huge sequence space of possibilities, and random mechanism selecting the right sequence. It is natural mechanisms. They are too unspecific to select the right sequence out of many possibilities, and the possibilities is what I am pointing out to you now.
All right, hold on. We got a super chat. I want to read the super chat before it goes away. So for $20, Mr. Wilford says, "Can I test your model?" Yep. And result X would be bad for your model? Yep. So if I show you result X in an experiment, then your model is falsified. That makes sense to me. Yep. We've seen X in the lab. That's not my model. So that was a humorous one, but I want to point out a few things. Okay, so you previously said that if we found functional proteins, any of them, from random genetic material, that would falsify specified complexity.
Let me stop you right here. I think we have to define here something.
Okay.
First of all, are we talking about the sequence of the genetic information stored in DNA? That is a problem. But in prebiotic real scenarios, you didn't have that relationship between information stored in the genome directing the making of polypeptide amino acid chains and proteins. There was no such relationship. So what do prebiotic abiogenesis researchers look for? They try to explain how the first proteins emerged without the direction of DNA.
Okay.
Because there was no such a relationship between these two things.
Otangelo, I'm going to try this one more time. I'm going to give it one more go. And then we're going to switch to, let's just take some questions from the audience. Okay. I'm going to once again restate specified complexity as a hypothesis. And I want you to tell me where I get it wrong. Okay. Proteins require specific... I'm getting some feedback. Where's that coming from? Josh, is that you?
It might be.
We'll see if it is.
Let's see.
Am I still getting feedback? Nope, I guess it's Otangelo. Oh well. So proteins have functions which depend upon, for any given function, a specified shape and chemical activity on functional sites of the protein.
Correct, yes.
Okay. This is coded for by genetic material.
Yes, agreed.
Such function cannot come about through randomly assembled proteins based on random sequences of amino acids or RNA or DNA.
Yes, agreed. Yes, okay. I agree with you.
So, and these functions happen in or around living cells.
Yes.
Okay. So, in 2001, Anthony D. Keefe and Jack W. Sostak reported in the journal Nature, in a paper titled Functional Proteins from a Random Sequence Library, that when they used a library of 6 times 10 to the 12th random amino acid sequences, they yielded 4 functional proteins that were unrelated to any other protein based on current databases in 2001 of biological proteins. So they started with random proteins. A non-zero number of them were found to be functional in or around a cell. In fact, they specifically were ATP binding. Okay. Therefore, specified complexity, assuming that this paper is accurate, has been falsified.
Okay. Okay.
Is that true?
Okay. So
I would really like a yes or no.
Yes. Okay. I am agreeing with you on that one.
Okay.
So you agreed that when a hypothesis has been falsified, it has to be modified in order to be tested again. Okay.
Well, you would have to modify your predictions. So what is the new version?
So my prediction is that you remove the scientists of the experiment, and then you start with over 500 amino acids supposedly laying around on the prebiotic earth. Then you demonstrate how the 20 which are used in life were selected from the pool of over 500 amino acids. And then you have the pool of 20 amino acids which are actually used in life. Now, then you have to demonstrate how you select the left-handed from the right-handed ones, sort the right-handed ones out.
What is your modification?
Let me finish. Let me please finish. I think this is important to point out what the real life problem is here.
But you're shifting the goalpost. Irreducible specified complexity was a problem for evolution. Now it's a problem for abiogenesis. Okay, great. Maybe it's a problem for abiogenesis, but it's not a problem. I'm not even addressing that right now. But it's not a problem for evolution. Is that true?
Well, I disagree with that. I think it is a problem.
Okay, then you're going to have to come up with a new prediction in terms of evolution. What is the new prediction?
Okay. I like Depper to exacerbate the contrast and the problem, and evolution is not the best way to do that. I think abiogenesis is a much better model to demonstrate the contrast, because there you do not have evolution. You have just 2 possible mechanisms. On the one side, you have just a random selection by unguided forces on prebiotic earth, or eventually physical necessity on the one side, and on the other side you have intelligent design.
Okay, that's great.
So if we even take that what you exposed previously is falsified in the experiment that you said, so I am modifying now my hypothesis, and my hypothesis is that you have to demonstrate how.
Nope, that's not how the hypothesis goes. Start with what premises there are, then tell me the predictions. Start with your premises, then tell me your prediction, and then we'll see what we need to demonstrate. What are the premises of this new hypothesis?
The premises is that you have supposedly a pool of over 500 amino acids on the prebiotic earth. Then you have to demonstrate that there was a natural mechanism which selected 20 amino acids which are used in life. Then when you have selected these, you have to select the ones which are used in life, which are the left-handed amino acids. And when you have this pool, then you have to demonstrate how they could polymerize. There is no known, as far as I know, natural mechanisms of polymerization without the enzymes. And then you have to demonstrate how they could have lined up in the right sequence, which in the end would form a functional protein. And when you have that, you have just one protein. You need 1,300 functional proteins, each one with a specific function. And then when you have that prebiotically without enzymes, then you have to demonstrate the transition from a prebiotic non-enzymatic production by natural means to a production by cells where enzymes produce proteins by the known mechanisms, which is through the direction through a genome.
That doesn't sound like a prediction.
Okay. Okay. I made a prediction. I made a prediction that that is a real life problem, and if you can meet it by natural means, hey, then you meet my challenge and my burden of proof. If you can't, then the intelligent design is a better explanation by using eliminative
I'm sorry,
induction.
I was muted for a bit.
Oh wait, so I may have missed it. What was the prediction? And secondarily, even if, like, how do you then conclude that if you fail to demonstrate what you think should be demonstrated, intelligent design is necessarily better?
Well, I will not repeat everything that I said. If you did pay attention, then I draw a real life scenario on a prebiotic Earth where you have just amino acids laying around randomly, hundreds of them, and that is from where you have to start your narrative to explain the origin of life.
Here's what I'm going to do.
I predict that only intelligence is capable of doing the selection of the 20 amino acids.
All right, everyone but me is muted because this can't continue. We have to come up with some way to make this not rambling nonsense. Okay, here's what I'm going to do. Otangelo, you have 10 minutes to start to state a series of premises which, if true, will lead to a single prediction that can be tested. If you don't, we are moving on to Q&A from the audience. Now, I'm going to unmute you, but not Josh yet. I'm going to unmute you Josh after the 10 minutes, and I'm going to mute myself for the 10 minutes. So, here we go. You are unmuted. I am muting me, and once you see me muted, I will be hitting the start button on my timer.
Okay. I predict that intelligence is capable of selecting amongst a pool of over 500 amino acids, 20 amino acids which are used in life. And when you have these amino acids, then intelligence is capable of selecting the right-handed amino acids from the left-handed amino acids and just join and reunite 20 left-handed amino acids which are used in life, and then sequence them into useful proteins. And not just one protein, but about 1,300, which are required to make a first living cell. We know that because there is the smallest free-living bacteria known, which is Pelagibacter ubique, which uses about 1,300 proteins with about 400 amino acids in average each. So we have a real-life minimal organism, which is the question which we have. How do we get to that organism? By natural means either, or by intelligent design? And my prediction is that intelligent design is capable of doing the selection of all those things, because intelligent design has foresight. It can invent and make things for specific purposes in long distance purposes, because it can invent those things. It has foresight. I predict that intelligence not only can do it, I don't actually only predict that it can do it, but I can say that this prediction has been met, because we know by our own experience that we as intelligent beings, we know how to make machines, how to make production lines, how to make a selection of materials to have a specific goal, but we do not have any experience that natural mechanisms can do all these things. So I think that intelligent design is already confirmed by the fact that we know that intelligence can do all these things. So this should be, in my understanding, the default position by science, and then try to refute this default position by demonstrating that natural mechanisms can do all these things, which is starting from a pool of about 500 amino acids existing on a prebiotic Earth, selecting those 20 which are used in life, selecting the left-handed ones which are used in life, then polymerize them to functional proteins, select a pool of about 1,300 proteins which can interact with each other and form a functional proteome, and then also generate the metabolic networks where they work in a joint venture together, and to form in the end a factory which makes the basic building blocks of life and starts to self-replicate, and which has all the features of life, which is to import materials, raw materials, process them, generate energy, and generate information, and then to go from prebiotic, non-enzymatic production to transition to enzymatic mechanism where enzymes actually produce all these things. And we know in over 70 years of scientific experiments that no one has been able even to demonstrate how the basic building blocks of life could have been generated abiotically. So we have in 70 years not made any progress whatsoever. What science has demonstrated is that actually the huge task which is in the realm of the absolutely impossible, to have a genome, a minimal genome, to have the 1,300 proteins, you require about 1.3 million nucleotides, and that if you ruffle that, if you see how what the chance is, it is about 1 to 10 to the 722 22nd. Sorry, it's 1 to 10 to the 722,000 power. That is in the realm of the absolutely impossible. So I think that if science would be honest in regards of this issue, it would have long ago come to the conclusion that natural mechanisms are simply not an adequate explanation to explain all these things which have to emerge prebiotically. And we know that intelligence is a more case adequate and more plausible explanation. Well, I've not used my 10 minutes, but I think that draws the picture. Okay?
I thought that's okay. So since I didn't hear any premises, you have, and the general consensus of the chat is very much that you did not actually manage to state a hypothesis. I am unmuting Josh, and we're going to go to some questions from the audience, and we're going to do that for a little bit.
Well, Dapper, let me just correct you. I think
no.
let me correct you on this. I gave you the hypothesis very clearly. I said all the things.
State just your premises. You got. I'll give you 2 minutes. Go.
Okay.
Just the premises.
The premise is that natural mechanisms are actually no. Sorry.
Okay.
The premise is that intelligence is capable to select 20 amino acids from about 500 occurring naturally on the prebiotic earth, select the left-handed amino acids which are used in life from the right-handed ones, intelligence is able to make chains of, join these together in a meaningful sequence which will permit them to have a functionality, and then make that process 1,300 times to have a proteome that would be a park of machines which work in a joint venture.
Okay, we don't need the full spiel on biochemistry is complicated. What is a testable prediction that could falsify those premises? One prediction.
One prediction. Provide a demonstration that nature is able to sort out right-handed amino acids from left-handed amino acids. That's called the homochirality problem.
Okay, so your prediction is that it is impossible for nature to solve the homochirality problem.
Yes.
Okay, since it's impossible to demonstrate a universal negative, do you have a prediction that is actually falsifiable?
Yes, we know by experience that intelligence is able to sort out if on the right side you have non-usable monoblocks, and on the left side you have usable monoblocks, that intelligence is capable to do that selection process and sort out only those blocks which are usable and can be used in a forthcoming construction site.
Okay, well, that wasn't a prediction. That was simply saying that intelligence can do a thing. I'm going to give you one further minute to make a prediction that we could test and falsify if it's not true. And at that point, I will not let you make further corrections. We are going to questions, which can include questions for you. So here we go.
Sure.
1 minute.
Sure. Okay. I think that the framework of making predictions in science is moot in regards of what I just said, because...
All right, you're muted now, Otangelo, because you weren't even trying. So I'm going to unmute you in a second. Just want you to know, complaining about falsificationism, that's great, but it wasn't the question asked. We've already long established that falsificationism is the only viable way to proceed in science. So now, anyone who would like to put question marks, please tag @DapperDino, so that way I see them. Super chats, of course, will go to the top of the list. We'll be here for probably another 25-ish minutes, and then we'll do some wrap-up, and then I will be out of here. So, Otangelo, you are unmuted. Sorry, but you had to actually perform the requested action in the minute, and you started off by not even trying. So,
well, I just had an idea now. I can give you.
We have a question from Beethoven for you, Otangelo, and it says, "Are you for real?" You don't have to answer that if you don't want to. I personally think you are.
Well, I would like, if you give me the chance, I would give you. I just had in mind an example where we can attest what you actually said.
Otangelo, I gave you somewhere around 13 minutes to try to do that once, and you didn't. So I'm not going to give you another chance. If you want another chance, here's my recommendation: make a video, or write down an essay or something like that. Send it to me so you know I know about it. And then I will examine it, and if it is worth responding to, I will respond to it. And either way, if you want me to, I can post a link to it or something on my community tab so that everyone can see your suggestion.
No, I think it is not necessary. I think I wrote through my point.
Okay. So Scott Duke asks, for Dapper Dino, can we nominate you for the first Atheist Angel? Only if you can prove I'm an atheist. Good luck with that. Yeah. Everyone's making assumptions. Let's see. Beethoven, this isn't a question, but I feel like it's worth reading. Plus, he's got that golden top hat. It makes him really fancy. He says, this feels fake. I think he's a Poe. Now, I recognize that the only answer that anyone typically gives to the question is no. But I guess I'll ask it. Otangelo, are you a Poe?
No.
Okay. Fair enough. I mean, that's the thing is, even if you were, that's the answer I would expect. So it's kind of like it's kind of like, are you a secret spy? Of course, the answer is no. What other answer are you going to give? Let's see. This is from Matt Janssen, I think. I don't know. I'm bad with the diaries. Diarist? Yeah. I don't know. Josh, you do some German. What's your take on that? The pronunciation?
I believe it's Johnson.
No, there's got to be a Y sound.
Oh, maybe.
Anyway, so he asks, he says, ask him, which I assume is Otangelo, is math descriptive or prescriptive?
Math is both. It is, you can describe it if you observe it, but you can also invent it, and then it is prescriptive.
Okay. Jerry Penna says, Otangelo missed my point. The point was about abio research dollars. Processes that happened a billion years ago have a fraction of the research, yet you expect all answers to abiogenesis.
Well, we have enough answer to know that it is not possible by natural means.
Okay.
I think you missed one above Matt. Yes, Bendit.
Oh, you're right. I did, actually. I'm sorry. So S. Bendit asks, Otangelo, how does intelligence sort chirality?
Well, I give you building blocks which which are usable in your construction site, and I give you homochiral construction blocks which are not usable in the construction site. And I, as an intelligent being, I can sort out those which are useful, which I can use, from those which are not. We know by experience that this is possible. So that's how.
Okay, and we have a $10 super chatted question from Milo Island. Otangelo, I'm sorry, sir, but you seem to get lost when you talk. You start with a premise, then halfway through you start to talk about random stuff. Can you define premise?
Well, the premise is let me think about it.
Okay. I'm going to look to see if there are any questions that are for me while you work on that, and then let me know. And
Okay.
Oh,
Yeah, there's another one from.
No. I can answer that one.
Okay, go ahead.
Well, that is actually the claim which is being made, and then you try to falsify it.
Well, that is not quite true, but okay. So a premise is a statement that is, in the context of the argument, assumed to be true from the outset, and then a series of these premises can be strung together with a conclusion saying therefore, and then that is the claim. So a premise might be all men are mortal, and another premise might be Socrates is a man. Then the conclusion is therefore Socrates is mortal. Now, I don't suggest this, but the Athenians bothered to test this hypothesis, and it turns out that yes, in fact, Socrates was indeed mortal, because it turns out that after drinking poison, he died. So,
well, I can answer to this. I mean,
well, we're going to move on to the next questions because
okay.
we're doing questions. Okay, so let's see. So Cheshire Vic asks, "Can I have a shot at this guy sometime with a sort of evil grin emoji?" So, Otangelo, how would you feel about being on a program with Cheshire Vic?
Sorry, I didn't understand the question. Sorry.
So Cheshire Vic says, "Can I have a shot at this guy sometime?" So she's asking if you would be open to having a conversation with her.
Oh, yeah, sure.
Okay. Mm-hmm. Would you like me to get her your email?
Yeah.
Okay.
No problem.
So Chesh, I will be getting you Otangelo's email, and if I don't know, you can come beat me up or something. Let's see. So, Ugly Truth says, at Deverdino, well, how many gods do it says, how many gods do believe in your existence? I guess if you're asking how many gods believe in me, then I presume if they're worth being called God, all of them that exist would probably believe in my existence, assuming my existence is real and not just an illusion, which I tend to accept. So I guess all of them. And if the question is how many gods do I believe in, then the answer is you'll just have to wonder. Yeah, it's mysterious. Yeah, I don't talk about my personal belief in gods. TP Seeker asked a question, but I don't think I'm going to read that particular one. Okay, so Silver Harlow asks, question for Otangelo Grasso, although he said OG, but it's your initials, so I figured it's you. Can we stop pretending 70 years is a long time?
I think it is more than enough time to make very secure conclusions.
Okay. Let's see. This is from Jerry Penna. What's Otangelo's goal? What does he want? Us to abandon abio research and accept Jesus?
Well, I use in my apologetics a cumulative argument for God's existence, where the first question is there a God or is there no God? And one of the good tools is to use the teleological argument for God's existence, which is my main focus.
Okay, so I'm going to ask that question again, and I would really like for you to answer it. What is your goal? What do you want for us to abandon abio research and accept Jesus? And that is a yes or a no. Just the last one. Would you like science as a whole to abandon abiogenesis research and for all scientists? Actually, we'll set that as a separate one. And would you like all scientists to accept Jesus? I assume the second one is yes, but...
Well, one thing has nothing to do with the other, but
That's
no,
fair.
I mean, I think it's okay to continue the research because it will exacerbate the impossibility of natural mechanisms to be able to, or being capable to bring forward life.
Okay. Let's see. S. Bendit says, you did not answer my question. If you gave a solution with two isomers of the same molecule, how would you separate them? And I think by you, he means the intelligence that you're positing, but maybe not.
Well, God is a super intelligent being. He knows which isomeric configuration will be functional and which one will not. And using intelligence, he will be able to sort it out, while non-intelligent mechanisms do not have that capability.
Okay. So if I say, how would an intelligent person separate vowel letters from consonant letters in a block of lettered blocks, right, like a kid's toy? My answer would be, using pattern recognition and a previous knowledge of the alphabet, he could sort them by hand, placing the vowel letters into one bucket and the consonant letters into another bucket, and keeping Y and W in a third bucket because sometimes they're weird. You just restated that an intelligent being could separate things using intelligence. What
Correct.
physical thing is going to happen inside this solution? Physically. I want you to tell me about where the chemicals are going to go, what filtering mechanism is being used, what's physically going on in there. And if you don't know, that's fine, but just say that.
Okay, that is a different question.
No, it's not. That is the question.
No, it is a different question in regards if intelligent or non-intelligent mechanisms are the most plausible ones. Then you are asking, but how did the intelligence do it?
He didn't ask which was plausible.
Asking. Well, usually it goes like this. When I point out that intelligence is the better explanation, then they move on and ask, well, but how did God do it? That is already another question, and I can give two answers to that. One is, in order for one explanation to be the best explanation, I do not have to answer other questions in regards of this.
Do you know the mechanism?
Well, we do not know the interface of physics.
I'm just going to go ahead and say that no, you don't know the answer because you haven't given it. And so,
I don't.
Okay, there we go.
Know.
So now we have from the Venerable Bede. Does he not understand that computers and factories don't reproduce and so cannot cannot be fill-ins for cells and nucleotides?
Well, that is a good question. I love it because reproduction is actually the epitome of advance of manufacturing, and if we as humans would be able to make self-replicating factories, that would be far, far more advanced than anything that humanity has ever invented in regards of manufacturing. So that is actually excellent evidence of design.
Okay. So Icarus says, oh, and by the way, I'm not planning to respond to your questions unless I feel like you haven't answered the question. So does Otangelo Grasso believe in dinosaurs, and how do they fit into the flood story? And this is from Icarus.
Well, of course, I believe in dinosaurs. I believe they were created by God.
And how do they fit into the flood story?
Excuse me?
How do they fit into the flood story?
I believe they existed prior to the flood.
Were they on the ark?
Probably a few of them. I don't know.
Okay. So let's see. Sexy Calzone asks a question for Otangelo Grasso, but he forgot to tag me, so he's lucky I noticed it. How does negating evolution or abiogenesis give us, maybe lead us to theism?
Well, there are basically two possible explanations. Either there is a God or there is no God. If there is no God, then of course natural mechanisms explain our origins. But if there was a God, then and natural explanations do not cut the cake, then of course God as an intelligent, super intelligent being explains our origins in a more adequate manner.
All right. Let's see. Philip Cordis, I almost missed one of those L's, or so those I's, and I thought it was Flip Cordis. Sorry. Philip Cordis asks, what probability do you find acceptable to accept random events can happen? Because you said 1 times 10 to the 50th was impossible. So at what point is it acceptable to say that this could happen randomly?
I think it is absolutely not possible in any sense.
What? You don't think anything randomly?
So nothing random can happen?
I believe that random mechanisms are an inadequate explanation at all.
For anything?
Well, for the origin of life and for
that wasn't the question. Okay, let me restate but I'm going to ask the question again, and we're going to try and get you to answer this question. What probability do you find acceptable to accept random events can happen?
I don't know. I would have to think about that question.
But 10 to the 50th is too high.
Absolutely. I mean, the threshold of Borel's law is 1 to 10 to the 50th power.
Okay.
So basically, the threshold is if something is less likely than that number, then it is in the realm of the impossible. But this is, of course, also an arbitrary thing.
All right. Ian Chen says to me, of course you're an atheist. Dinosaurs never developed religion, to which I would say, I'm not sure how you could know that, because I don't remember you being around in the Mesozoic era, Ian. But plus, I've been around for the whole Cenozoic, and some mammals have managed to develop religion, and who knows, maybe I liked it. Let's see. We have from Zach Brannigan. Ask Otangelo, is there anything in the Bible he believes to be false? So is there anything in the Bible that you believe to be false?
No.
Okay. Let's see. Scott Duke says that he has to go. Thank you very much, Scott Duke. Especially thank you for the super chat. Let's see. Let's see. This is from... I'm still going to go with Mats Jonsson. Can Otangelo falsify magic?
If I can falsify magic, well, I would have to ask first, how would that be?
Okay. Is that a no?
Yeah, leave it by that.
Okay. So Ugly German Truths says, somebody mentioned in the chat that Otangelo Grasso believes the smallest extant cells are similar to protocells of the ancient past. Can you ask him, please, what he thinks the smallest cell is?
I think the smallest cell is a free-living, self-replicating cell, and we have an example, which is Pelagibacter oblique. It has 1.3 million nucleotides, about 1,300 proteins, and about 1,300 genes. I think this is the threshold. Many say mycoplasma, but it doesn't make amino acids by itself. It has to take them as nutrition from the outside, from the host. So that is, in my understanding, not the threshold of the minimal life.
Okay. S. Bendit asks, Otangelo Grasso, if you cannot separate isomers, is it because you are not intelligent or because of some other factor?
I said that we as intelligent beings, we can separate useful things from those which are not useful, but a random action is not capable to do so.
Okay, I'm going to ask the question again. If you cannot separate isomers, is it because you are not intelligent or because of some other factor?
It can be for some other factors.
Okay. So let's see. You know what, Josh? I'm going to start using this technique with you. I'm just going to repeat the question until you answer the question I ask.
Sometimes I misunderstand the question.
I know. I'm sorry. Just taking a quick dig at Josh. Let's see. This is from Jerry Penna. How does Otangelo account for the 1,300-year gap in chain of custody in the Shroud of Turin? And actually, before I start with this, are you someone who accepts the validity of the Shroud of Turin as the actual burial robe or shroud of Jesus?
Absolutely.
Okay.
Yes.
Okay, so then how do you account for the 1,300-year gap in the chain of custody in the Shroud of Turin?
Well, I have an entire article at my library. I can link to it, or you can do it, and then they can read about it.
Do you have maybe like a 2-sentence summary so that people know what they're getting into?
Well, there are several explanations why they came up with that dating. One is that the piece of cloth...
No, he's not asking about the dating. He's saying for 1,300, because this appeared in the 1300s, if I'm correct, right? That was
no, it appeared previously. There is history before 1300.
Okay, so Otangelo's answer is roughly that there really isn't a gap, and okay. I would encourage you, what is your website?
reasonanscience.catsports.com. Eventually you can link it, or I can put it in the chat afterwards about that one.
I'll tell you what, actually, put it in the private chat, and I will add it to the description of this video, and that way anyone can go there. So I will add Otangelo's website, and then as soon as you get that in the private chat, I will copy paste it in, and we will have a link there.
It's already there.
All right, let me copy that link address, and there you go. So that has been added to the description of this video. So feel free to head over there. Not yet though. You gotta wait for this stream, which is going to be done in about a few minutes. Let's see. I think this is going to be our last question, unless we get a super chat. Let's see. This is from Silver Harlow. It took 240 years for Einstein to find an improvement to Newton, and that was a trivial compared to abiogenesis. Can we get at least 240 years before we give up on abiogenesis?
It's not necessary. We know already that it is impossible by natural means, and that is very, very clear by what we already know.
Okay. And then I'm just going to go ahead and say, from Sexy Calzone, this isn't a question, but I think it's nice to read. And he says, comment, sorry, for Otangelo, I know I give you crap in the chat, but good on you for not being someone who refuses to go outside their echo chamber. You're my favorite ID proponent. So I just thought that that was a nice thing to have in there. Yep. Yeah.
You did skip someone, though.
Yeah, that's because I said we were doing the last question, unless there was a super chat. Because we're wrapping up.
Right. Gotcha.
All right. So, Josh, do you have anything that you want to plug? And if so, plug it and then get out of here.
I have nothing to plug.
All right. Otangelo, what would you like to plug? Websites?
Yes, well, you've put it, I think, already. It's Catsport. My two channels on YouTube. One is The God Talk. The second one is Intelligent Design Academy.
Okay. And also, I linked to both of the Otangelo Grasso channels that I'm aware of on YouTube. I know you have 2 accounts using that name, and also similar but not identical profile pictures. So those are both there. So if you would like to, you can subscribe to Otangelo Grasso. Do you have any other engagements coming up that you already have scheduled that you'd like to tell people about?
Yeah, yes, this weekend probably I will have a talk with Dimitar Kunev. He is an abiogenesis researcher. We have not set yet the date, but it will be on my channel, The God Talk.
I don't think I've linked to that one. I will try.
Yeah, I've not set the date. I can give you the link to the channel. You can put it there, but
I'll add that.
We have not yet agreed on the date exactly. It will be this weekend, but I don't know exactly at what time.
Yeah.
It will be tomorrow or
I'll
Sunday.
just link to the. I'll add a link to the God Talk channel. It might end up going in after the end of the stream, guys. So check back in a little bit. It'll be there. So we also have a $2.99 due super chat from Ian Chen, who says, super chat. All right, so the next thing you're going to get from Dapper Dino is Kent with Bent 50, and unlike the NFL, I have confidence that my audience is smart enough to know what the letter L represents in Roman numerals. So we are going to be putting Kent with Bent L in the thumbnail, because I know you guys are smart. Not that football fans aren't smart. I actually think the NFL underestimated the intelligence of their fans, and it bugs me. So I am not going to patronize you like that. And then Thursday, I haven't decided what's going up there yet. It's probably going to be a re-upload of my debate with Otangelo here, so that way I have a copy of it on my channel. And I understand if you would want to skip that, but hey, it was a fun chat. I enjoyed it. And that is basically it from me. So, Otangelo, I want to say thank you very much for coming on. And at the very least, we didn't end up in a shouting match, which I have seen some conversations like this end up in. And I thank you. And I hope that your conversation with, it's Dmitry, right?
Dimitar, yes.
Oh, Dimitar, okay. Sorry.
Yes, he has been already sometimes at my channels, but I like to have polite conversations with atheists, and I think it was a nice conversation with you guys, and thank you for having me at your channel. And
Otangelo, you don't get to say atheist. Only one person here is a confirmed atheist, and it's not me.
Sorry. Yeah, okay, sorry.
Okay, that's one of my sticking points. You don't get to call me an atheist unless you know it's true.
Oh, okay, sorry, I apologize.
That's all right. I'll just end with, guys, please, if you enjoyed this, give it a like. If you didn't, hit dislike. Please, I encourage dislikes. If you didn't like this, put a dislike. Let me know what you didn't like. And other than that, if you haven't already subscribed, please do. And also, as I said, Otangelo's links are either, well, many of them are currently in the description, and one more is about to be added. Thank you very much, and I will hopefully see all of you and more on Tuesday.